Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last two classes, we have started the hands on and we have set up the system in R. Okay. And very briefly, we discussed about the common commands and we have looked into some of the plotting tools, how we can plot data and generate very nice plots of the data. Okay. We have also seen very basic statistical tests that we can do with R. Okay. So, there is a lot of things to explore which we cannot possibly cover in very few classes, right. So, I will urge you to explore yourself, right, and you will see like this is a very powerful programming environment where you can generate a very interesting plots, very complex ones. Also, you can organize the data or organize the plots in certain ways that the ways you like, and you can perform all sorts of statistical tests. So, coming back to our analysis, the agenda for today's class is that we will go into the preliminary data analysis part. Okay. So, this is what we are going to discuss today. So, again reminding you all the steps, we have set up the system in R, we have looked at the commands very briefly, we have loaded the count data, right, with 16 samples and about 6,800 genes, and we are now going to go into the preliminary data analysis. Okay. Just to remind you, we talked about two types of preliminary data analysis when we were talking about the this in theory, right. So, we have this distance based clustering we generated. Again, if you remember, right, we have these 16 samples that are present in 4 groups. Okay. So, the groups are given here G1, G2, G3 and G4. Okay. And the distance are shown by these color codes here, right. So, if the distance is 0, you will see this very dark red color, and lighter color or white color means they are far apart in terms of their gene expression pattern. Okay. So, this kind of helps us visualize the whole thing very nicely. Okay. The second type of analysis that we did or that we mentioned in the theoretical theory part, right, is that principal component analysis. Again, this is a very very briefly, this is a dimensionality reduction method, right. So, which actually generating these principal components from this 6,800 genes, or it can be even more depending on your data, and then it kind of calculates the variance, right. So, that are explained by each of these principal components. Okay. So, here we have these two principal components we can see, and we saw like we can isolate these 4 group 1 samples, right, they are separate from the rest of the groups. Okay. And we also noticed that one of the group 4 samples, right, is actually stays far apart from all the data that we have. Okay. So, maybe there is some issue with preparing the library or during the sequencing with this sample. So, what we want to do now is, given our data, we want to generate this kind of plots. Okay. We want to do this distance based clustering, right, and then also we want to do this principal component analysis with R and generate this kind of plots that will help us visualize the data, and then will help us, right, if we want to maybe remove this replicate of G4 from our analysis because this is an outlier, there was some issue with this sample. Okay. So, let us now move on to R and then write the codes to actually do this kind of analysis. Okay. All right. So, we move on to R here, right. So, we have the data. To do this, we need a lot of packages, okay, and there is a pipeline that we will have to follow. Okay. So, this is something again what I will do is I have collected all the codes, I have developed the scripts that I will show you how this works. I will also mention the packages that we are going to use. The question you might have is that how do you find this out? Okay. So, this is something that is actually well known. If you are working with these packages for a long time, you know which package can do what, right, and then of course, you can learn, right. You can go through these packages when new packages are coming out or they are published, right. You will see in certain publications they are discussed, right. You will actually learn about these packages and then you can utilize those packages for your analysis. Okay. So, what I have done is I have compiled a code, right, and I will go through that code one line by one line, right. So, that we actually understand what we are doing, right. I will also show you these packages very briefly and what they can do. Okay, so I will clear this plot. So, to clear this plot is called dep.off, right. So, it will remove, or it will kind of close this window from here, right, because this is not relevant anymore. We will generate new plots that will appear here. Okay. So, you can see this actually is gone, right, and you can clear the window and we can start afresh. Okay. So, now we move on to the count data, right. So, that is the data we will work with before we actually do the dsec2 etc., right. So, the count data is inside this test directory, right, and we are here, okay, and inside this we have this run 3 all S1 S16 analysis. Okay. Just to very briefly I just open it again, and you can see we have these samples S1 to S16. Okay. S16 you cannot see here, it is at the end, right. So, we have 16 samples and we have these 6,800 genes. Now, the question is, where do these samples belong, right? What are these replicates, what do they mean, etc., right. So, the experimental design would also have to be mentioned before we actually go to any differential expression analysis, right. So, the program dsec2 would need to know, okay, which sample belongs to which group or which condition, right, and which are the technical replicates of one another. Okay. So, is S5 a technical replicate of S1, is S9 a technical replicate of S1, or S13 is a technical replicate of S2, right. That information would have to be given to the program, right. So, to do that we also have another file which is called this sample information file. Here you can see this sample information file, okay, and we will load this sample information file, okay, and you will see this very easily now. Okay. So, in the sample information file you have two values, right, two column names, these are headers again. Okay. So, the first one is sample, okay, this is the sample name we have mentioned in the count data file. Okay. So, we have S1 to S16, again they are arranged in certain order because of their groupings which are given in this column bin. Okay. So, in the bin column we have these groupings given. So, the S1, S5, S9, S13, the first 4 samples they belong to group 1. Okay. So, this is mentioned here. The next 4 S2, S6, S10, S14 they belong to group 2. Then we have S3, S7, S11, S15 they belong to group 3, and the last 4 S4, S8, S12 and S16 they belong to group 4. Okay. So, this bin actually gives the information about the experimental design. Okay. And we will see, we will use this information when you go for differential expression analysis, right. So, this is important because now the program will know, okay, these are the replicates, technical replicates of each other, and these are the different groups that we want to compare against each other. Okay. This information is very important. Okay. So, the bin part is very important. Of course, you can have multiple columns, you can have other components of this experimental design as we have discussed, right, when we are discussing the theory of this differential expression analysis. You can have, for example, age of the population or the sample, right, and you can have other parameters, right, and you can combine those in the linear model. Okay. But for now let us focus on this, right, where we have this very simple design, 4 groups, okay, and each group has 4 replicate measurements. Okay. So, this information would also have to load in the R program, right. In R console we need also need this sample information. Okay. So, for this preliminary analysis what I have done is I have developed this R code, as you can see, prelim analysis R code.R. So, inside that we have this code which will run, and of course, I will explain the commands that we are using. Okay. So, to see this I will just simply run this. Of course, we can run this R code as a script, which I want to, because I want to explain all the steps that we are going through. Okay. So, do not pay attention to everything that is there, I will go one by one. Okay. So, the first library here that we need is the dsec2. Okay. So, we have probably loaded this, but I will again load this here, library dsec2. We can put it inside double quote, or we can simply say library dsec2, it is okay. Okay. We do not have to worry. So, it is loaded now because it was already there. So, it is there now. Okay. So, we have to load this data and sample files, right. So, this sample information files in our console, okay, and we will do that one after the another, right. data equals to read table, okay, and header equals to true, right, because we have the header which is the sample name, right, and the data is loaded now. So, we can simply check whether the data looks fine after loading. You can always check this, right, when you are loading any data, etc. So, you can see this here that this is fine. We also will load this sample information file that I just showed you, okay, and it is loaded by this sample header equals to true. Okay, so again we have the header, remember, right, we have the sample and the bean, that is the header, right, and you can again check head sample, and we have these columns sample and bean. You can again explore, right, sample bean. So, it is telling me the information that is present in this column, right, and we can also try maybe summary and see if this gives us any information, right. It just gives you some statistics, that is really not important. Let us see if we can get the level information from here. No, it is not. Okay, so this requires a specific data format that will give you this level. Anyway, so we have loaded this sample information. Now, the next step is to convert these as data frames, right. So, keep this as data frames because the sec2 actually requires them as data frames. Okay. So, to convert that, we simply write as data frame, right. So, in R, this is actually possible to convert one type of data into another, okay, and this is what we are doing here. You can see this in the command. So, I am just writing data matrix, right, and this will be utilized later on. Okay. So, data matrix is as data frame, sorry, there is a mistake here, right. So, as data frame, okay, and simply just say data, and similarly we can convert this sample to also sample matrix. Okay. So, these are now converted to data frame because they will be required, okay, for this analysis. Okay, so what we are going to do now is we are going to convert this data into something called a DSEG data set. Okay. This is very important. This is the first step for doing this differential expression analysis using DSEG2. Okay, so we can now check this DSEG2 package, okay, and this is what you will see. Okay, this is again the bioconductor page for DSEG2. We have the manual here, okay, and just opening the manual we will get this all this information. So, before you actually go about doing this differential expression analysis, you have to create something called a DSEG data set. Okay. Now, DSEG data set, it combines this count data along with the sample information, right. So, this information is important for all these comparisons, right. So, that because it contains this technical replicate information, the condition information, right, the design of the experiments. Okay. So, that combination we have to create. Okay. So, that part we have to do through this DSEG data set class. Okay. So, this is where we have to create this data set from our count data that we have loaded as well as the sample information. So, this is something you will see in different R packages that they require certain data format before they can process the data. Okay. And we have to first convert this count and all this information into this specific data format, and here it is, right. So, you can see this is how we can convert this, and the command is DSEG data set from matrix, right. So, the matrix that we have created, we can now convert these to this DSEG data set. Okay. So, that is what we will do. This is the command, and as you will probably notice that there are multiple commands that you can run from this DSEG data DSEG2 package, and these commands, right, these are given here. Okay. You can see these commands that are given here. There are a lot of things that are here. We will not discuss everything right now. Okay. But you can also just click on these links. So, these are also links, right, with the page numbers. So, you can simply click on these links and you will go to this page, okay, where all this information will be there, the description of the function, the usage, right, how it will be used, what are the parameters it will take, what are the arguments it will take, right. So, and the description of the arguments, right. So, this will help us understand the kind of data that we need to feed to this program or this command, okay. And then of course, there are a lot of details and sometimes also some examples, okay. So, here are some examples, right, how you can actually use this command here. Okay, so, for now it is enough to know that we need to create this DSEC data set from the file. Okay. So, from the matrix we will create this data set and this command is this. Okay. So, again following the manual. Okay. So, we have to create this DSEC data set, we store this in this variable DDS DSEC data set from matrix, that is the command inside this DSEC to package. We first need the count data, right, this is present in the data matrix, right. Then we have the column data, right, that is in the sample matrix, right, this gives us the design of the experiment, and then finally it tells us, okay, the design, right, how this experiment has to be interpreted. Okay. So, design is the bin column, right, this bin column gives the information about different conditions or the different types of samples that we have, okay, as well as the technical replicates. Okay. So, this is the command that will run. Okay. So, let us run in R now, okay, DDS. So, let us clear this so that you can see clearly. So, even though we clear the window, this information is present, right, it is not just gone, okay, everything is there, the package is loaded now. Okay. So, DDS is DSEC data set from, again you have to be careful about this capital letters and the small letters, right, they are all mixed up here again. So, that is why you have to carefully look at the commands. Okay. So, the first thing we need is the counts data, right, that is present in data matrix. Then I think we need the column data, right. So, again we have to carefully look into how we should specify, right, sample matrix and design equals to, I think, right, bin. Okay. Again I have to be careful whether the bin is in capital letter or in small letters. Okay. Again it will look up in the file, right. So, the sample information file you can very quickly check the bin is in small letter, right. So, we can simply change that now. Okay. So, we have now loaded this, right. So, we are specifying the count data, this is the experimental count data that we got. We have the column data, this is containing information about the experimental design, and the final design equals to bin, right. This is telling, okay, interpret the design by using this bin column, okay, that contains information about the replicates and the conditions. Okay. So, it is giving me some error, right. So, let us see, okay, what we have. Okay, let us see. Oh yes, so I think I have made a mistake. So, one of the commands is not counts, it is actually count data. Okay. So, this part again you have to be very careful, and this is again we have to pay attention to this package manuals, right. So, where you have this DSEC2 from data set, right. So, let us again have a look, right. This is something that will be mentioned here. Okay. You can see this is counts, right. So, this is CNTS. So, if you have to mention this as count data. Okay. So, that I will change that and then it should be fine, it is not just count. So, in some packages it will say it will counts. So, again you might get confused, and now it is fine. Okay. It is giving me a warning message, but that is okay, right. So, it is saying like some design formula characters, so it is converting them to factors, so it is okay. So, the warning is fine, it is able to read the data. I can check, okay, whether it has converted this matrix data into DSEC dataset format, and it has, right. So, you can see this if you just type DDS, the class is DSEC dataset, right. So, this is the dataset format that has been created. So, DIM, this is the dimension. So, 6805 rows and 16 columns. Okay. Then you have the SS, that is the count data it has, right, only the raw count data. The row names are the gene names. So, this is mention, right. Of course, it will not give you all the gene names, but you can see these are the gene names. So, row names such as gene names. Then you have the column names. So, you have 16 columns from S1 to S16, okay. And then the column data names too, because these are the part of the design of the experiment, okay. So, this is fine now. So, we have converted this data into this DSEC data set. The next step, okay, is to convert or to transform this DDS data through this R-log transformation. Okay, so again, without going into a lot of details, again this is a transformation that will convert the count data, okay, into certain format, right. So, it will take a log and etc. It will transform. So, this R-log transformation, okay, will then use for doing this preliminary analysis, okay. So, let us run this. So, RLD R-log DDS blind equals to false, okay. So, again you can check where this what this R-log means, what are the commands that we have, but for this preliminary analysis we need this R-log commands, and this comes from our default package of R, okay. So, this has run now. You can see this R-log is done. So, this transformation you can again check this head gives is the so head, again using this head command, right, you can check this the data, okay, the RLD data or the R-log transform data. You can see the counts have changed, right, because it has been, they are smaller because there is a log transformation, right, that has been done on the data, okay. So, this is the data, and now we want to do this limits, okay, and why we are setting this limit, because we want to check the correlation between these different technical replicates, okay. And you probably have seen this already, this correlation, and here it simply says plot assay, right. Some of these part are probably familiar to you, right. You can see assay RLD. So, this is actually looking at the count data only, okay. RLD has you can check what are the components of RLD by just typing RLD. Then it is saying 1, 2, again now we have talked about this is matrix, right. So, we are accessing the first two columns of this matrix 1 and 2. So, starting from 1 to 2, PCH we have talked about, this is the point type, then CX this is the point size, we are making it smaller because less than 1, main is the main title, we are changing xlim and ylim, we are setting the limits from -2 to 20. We have used xlim command right before. So, this should be familiar to you, okay. Once we plot, this is what we see, okay. So, what R-log does, it not only takes a log transformation of the data, it also does something called the variance kind of stabilization kind of analysis, okay. So, what is this variance stabilization analysis is that it's kind of minimizes this higher variability of this data of the genes that have low read count or low expression value, okay. So, it's very similar to variance stabilization analysis, we will not go into that detail, but you can see now this variability among these reads or genes that show low read count is gone, okay. So, it looks very nice now, there is a very good correlation in the data, okay. And similarly, now we can do a plot, right, we can add this y equals to x line, we can do a lot of things, okay, but we will not go into that, we have done this already. For just checking RLD, we can simply type RLD, it is a transformation that has been done. Again the dimensions are given, right, the row names are given, and then you have the column names that are given. And what has been done also is this size factor calculation, so that kind of normalizes this data, and this part of this high variability part is gone. Okay, so without going into a lot of details, right, so we can now see this very good correlation between technical replicates, okay. So, this by the way correlation between S1 and S5, right. So, we are looking at the first two columns of this RLD data. So, this is between S1 and S5. Now, what we will do is, now we want to generate this distance based correlation, right. So, this is what we will do in the next part. So, you can see this code, this we need these two packages. We will see if we have these packages installed, if not we will do them, and then we need to calculate these distances, right. So, distances between these transcriptomes or the expression patterns using Euclidean distance, okay, and then we will plot this using a heat map function. Okay. So, we will use this library p heat map to actually generate this distance based correlation, and we will also get a hierarchical clustering in that. Okay. So, let us see if we have these libraries installed, library p heat map and our library our color brewer. So, p heat map is the library that we need for generating this heat map, and the color brewer is a just giving us combination of colors. Okay. For defining these color palettes, etc. Okay. So, let us see if we have these libraries installed or not, right, in there. P heat map, let us see if it is installed. If not, okay, so it is saying that is okay, and also R color. Okay. So, these are now loaded here. Okay. What I will do is I will clear the window. I will also close this window, right, and now we can generate these new plots. Okay. So, let us look at the code again, right. So, what we will do first in this part is we will generate this distance matrix. Okay. So, between these different genes expression patterns, we first want to calculate this distance. Okay. For doing that, we will use this command. So, we will store this distance in this sample distance variable, right. So, that is why this assignment to this variable, we are calculating distance. So, using this command dist. Okay. So, this will calculate distance, and T is the transpose of this matrix SARLD. Okay. So, again if you look into how this distance will work, distance function works, we need to take the transpose. So, if you are working with this raw count data, right. So, or normalized count data, we use this Euclidean distance, which is the distance matrix. Okay. So, let us now run this in R, sorry, not here. Okay. And see if we got this here. So, the distance will be stored in this sample distance variable. Okay. So, you can simply write, and you can see this pairwise distance data. Okay. So, this distance between S1 and S5, distance between S1 S9, right, and you can see this matrix, only the lower part is filled. Okay. The upper part is not filled, and the diagonal is of course 0, because distance between S1 and S1 is 0. Okay. So, once we have generated this distance matrix, now we can use the heat map to plot the data. Okay. And we can actually use some certain color combinations, right. So, you can see we will use these color combinations to actually plot the heat map. Okay. So, we will get these row names and column names, right. So, again this will be used later on, right. So, we can get these row names, column names. Okay. And okay, sample distance matrix is not found. Okay. We will see if we can. Okay. So, okay. So, first we actually calculate. So, we have calculated the distance, but we need to convert this to a matrix, right. First we need to run this command. We have not run this, we have not created this sample distance matrix. Once we create that, then we can set the row names and column names of this sample distance matrix. Okay. So, we will use this sample distance matrix for the heat map generation, right, as a matrix form. So, p heat map will take the data in a matrix form and will generate this distance based clustering plot. Okay. So, that is why we are getting this error here. So, first we generate the sample distance matrix here. Again we are converting this data into matrix form. So, as I said, like in R you can do this, right, you can interconvert between different data types, and this part, the row name sample distance matrix, is we are setting the row names of sample distance matrix from this RLD $bin, right, and RLD sample, and with this separator. Okay. So, we will see when we generate this correlation plot or the distance plot, we will get this bin and the sample name as the labels. Okay. We will see this in a moment, and the column names of the sample distance matrix are null. So, we are just setting as null. Okay. We do not want any specific name there. Okay. So, now we can set the colors, right. You can, again this is for some very nice colors. If you want to change the colors, we are using this color ramp palette from this R color brewer package. Okay. And we are using, let us say, the blue colors, right. So, we are using these blues. Okay. You can check this, we are not explaining everything, right, how this works, but this you can check that this is from the our color brewer package, and this is simply for setting the color that we will use in the heat map. Okay. So, now we can actually use this p heat map command. Okay. So, I can go back and I can show you, right, our p heat map. Okay. So, where so our heat map in CRAN, right. So, where you will have this manual, right. So, CRAN package p heat map, and you will have this reference manual here p heat map .pdf, and inside you will have this command. So, this is this p heat map command, and the arguments that it can take. Okay. So, again you have this description of the arguments. You need a numeric matrix, of course. You need also a color argument, right, that you will give, and then other different types of arguments. Again, you can give them default or you can use them. Okay. So, what I have done is I have given this matrix data, right. This is the matrix data that we have to supply, and then we are saying like how you should cluster the samples and which colors it should use. Okay. So, the color equals to colors argument, right. This actually sets the color from this our color brewer, right. For other variables it will just simply take the default value. Okay. So, we can now use this command. Okay. So, we have set color equals to colors, we have given the sample distance matrix, right, and we have said like use these distance rows and also the distance columns from the sample distance list, right, and then generate the plot. And you can see, we see this very nice plot now. Okay. Very similar to what we have seen earlier, and what simply tells us, okay, these G1 samples, right, and these are the replicate or the sample names, right. So, S9, S5, S1 and S13, right. So, these are all part of this group 1, okay, and that they are present here, and there is a very nice correlation between them. Okay. So, these group 1 samples, they are very nicely correlated with each other. Similarly, for this group 3, group 4 and group 2 samples, you can see very nice correlation again among each other, but then you also notice that there is one sample here G4S16 that seems like an outlier. Okay. So, this is what we wanted, right, what we wanted to generate in the first place. Okay. So, this is how we can do this distance based clustering, right, which can tell us, okay, which samples are close to each other and which sample might be an outlier in our data. Okay. So, in the next class we will talk about the principal component analysis, right, and we will generate this principal component analysis plot, and once we are done, then we can move into the bias correction part. Thank you very much.